Welcome to my YouTube channel. The topic for discussion is physical methods of microbial control. Why do you need to control microorganisms? You need to control microorganisms to prevent transmission of infection and diseases. Also, you need to control microorganisms to stop food spoilage and also deterioration of materials. There are a few terms that have to be defined before we go on. Sterilization. The term sterilization is a process by which all forms of microbial life are killed. That is, at the end of the sterilization process, no microbial life should be found there, whether vegetative cells or spores should not be found on that object. So it is either the object or material is sterile or is not sterile. There is nothing like, oh, it's almost sterile, oh, it's semi-sterile. Another term is disinfection. It's a process by which infectious agents or pathogenic microorganisms are removed from the material, and it is important that bacterial endospores can still survive the disinfection process. Heat is a physical method used in controlling microorganisms. There are two types of heat treatment. You have moist heat treatment and you have dry heat treatment. The moist heat treatment ensures that microorganisms are killed by denaturation of enzymes and essential proteins, disruption of cell membranes, and degradation of nucleic acid. The various types of application of moist heat include boiling, steam sterilization, pasteurization, and tyndalization. The second type of heat application is dry heat. This involves flaming, incineration, and use of hot air oven. The moist heat treatment involves 1. Boiling. The material is subjected to the temperature of 100 degrees Celsius for 10 minutes. Boiling ensures destruction of vegetative cells and non-spore formers. The second type of moist heat treatment is steam sterilization. This is the application of saturated steam under pressure, and this destroys vegetative cells as well as bacterial endospores at the temperature of 121 degrees Celsius and pressure of 15 pounds per square inch for 15 minutes. To achieve steam sterilization, an equipment known as an autoclave is used. It could be the horizontal autoclave or the vertical autoclave. Now, it is important that before the use of the autoclave, the level of the water above the heater is checked, and the loading of the materials into the inner chamber is done in such a way that there is enough space for the saturated steam under pressure to penetrate. So overloading of the autoclave should be discouraged. Now, once the autoclave is covered, the valves are left open, and as the temperature rises, air is driven out by the steam, and after some time, the valves are covered, or rather closed, and the temperature rises to 121 degrees Celsius, and the timing is usually 15 minutes. Before the closure of the autoclave, it is expected that a biological indicator should be included. This biological indicator is used to check whether the sterilization process or sterilization protocol was complete. Now, what is it made up of? The biological indicator consists of an ampoule or a vial of culture medium, which is used together with a strip of spores of Bacillus stereothermophilus. At the end of the sterilization protocol, the strip of Bacillus stereothermophilus is inoculated into the culture medium and incubated overnight. Now, if no growth occurs after 24 hours, it means the sterilization protocol was successful. Another type of application of moist heat treatment is pasteurization. This is the use of moist heat to kill bacteria, pathogens, and spoilage organisms in liquids and beverages. Louis Pasteur is known to have developed the process known as pasteurization today. It was observed that the French wine industry had problems increasing the shelf life of wine that was produced. So Louis Pasteur took some of the freshly prepared wine and observed them under the microscope. I mean, they spoiled, not the freshly prepared, excuse me, they spoiled wine. He took some of the spoiled wine and observed under the microscope that some microorganisms were responsible for the spoilage of the wine. So he subjected freshly produced wine to a controlled temperature of 63 degrees Celsius for 30 minutes. And after that, he observed that there were no microorganisms in the wine that was pasteurized. Now, pasteurization was extended to milk because there are bacterial pathogens that can cause diseases in humans. These bacterial pathogens include Mycobacterium tuberculosis, which can cause tuberculosis, and also Brucella abortus. Apart from the vat pasteurization, another type of pasteurization is flash pasteurization at a controlled temperature of 72 degrees Celsius for 15 seconds. The fourth type of moist heat treatment that can be used is tyndallization, though an old method, but it's important to mention it. Tyndallization was developed by John Tyndall, and it's known also as fractional or intermittent sterilization. The materials to be sterilized, whether culture medium, have to be loaded into the equipment and subjected to heat treatment, moist heat treatment at 100 degrees Celsius for 30 minutes. Following this heat treatment, the materials are moved on cooling to an incubator set at 37 degrees Celsius for overnight incubation. Now, the vulnerable vegetative cells are destroyed during the process of tyndallization or fractional sterilization. But the heat-resistant bacterial spores will survive the treatment, the heat treatment at 100 degrees Celsius. So on incubation, they will germinate into the more vulnerable vegetative cells. So the second day, again, the materials are subjected to the moist heat treatment in the equipment at 100 degrees Celsius for 30 minutes. And on cooling, the materials are moved into the incubator set at 37 degrees Celsius for overnight incubation. These will enable the bacterial endospores to germinate. And on the third day, the materials are again subjected to the moist heat treatment of 100 degrees Celsius for 30 minutes. This is just a precautionary measure. Now, comparing sterilization and steam sterilization, it's obvious that you don't have to wait for 3 days to achieve sterilization when you can use the autoclave with saturated steam under pressure to achieve sterilization at 121 degrees Celsius in 15 minutes. Application of dry heat treatment. This ensures microbial death through rapid oxidation of cell constituents and protein denaturation. This includes flaming of wire loops, inoculating needles. These should be held like pencils in the flame and not in horizontal position. Now, you have the use of incinerators. You have the infected laboratory animals. They are best burnt. With pathogenic organisms, they shouldn't be thrown away like that in the refuse bin. Also, contaminated dressings should be burnt. An adaptation of the incineration in the lab is we have what is called bench incinerators, which can be used to flame wire loops and inoculating needles. The most important dry heat treatment is the use of hot air oven set at 160 degrees Celsius for 2 hours or 170 degrees Celsius for 1 hour. The hot air oven has a fan to distribute the heat evenly. Now, materials to be sterilized using the hot air oven include glassware, the glass petri dishes, metal instruments, forceps, a pair of forceps, pipettes, and others. Powders and greases can also be sterilized using the hot air oven. Low temperatures can be used to control microorganisms. In fact, the use of refrigeration exerts microbiostatic effect. What does that mean? The suffix static means inhibition of microbial growth. That is, microorganisms will stop growing, but that doesn't mean they are dead. It is important to note that not all microorganisms are killed in the refrigerator. You have psychrophilic and psychotrophic microorganisms that may spoil food at refrigeration temperature. The use of low temperatures also include freezing, lyophilization, which is freeze drying. This freezing can be done at -20 degrees Celsius and lyophilization at different temperatures, much lower temperature. And The ice crystal formation ensures disruption of the cell membranes of microorganisms, resulting in death. The third physical method of controlling microorganisms is desiccation or drying. This is simply the removal of water or moisture from a product, e.g. food products that can be dried include fish and even meat. Desiccation ensures that the water activity is low, and so microorganisms will not be able to carry out their metabolic activities. The moisture content of food is reduced. Microorganisms will not survive in such dried foods, except the halophilic, that is the salt-loving, and osmophilic, sugar-loving organisms. Filtration is another physical method which can be used in microbial control. There are several heat-sensitive biological products such as vaccines, antibiotics, hormones, and enzymes that cannot survive heat treatment. So they are made to pass through filters of known pore size. Asbestos and sintered glass filters were used to achieve filtration, but are now replaced with membrane filters that are made up of nitrocellulose and polycarbonate material. High efficiency particulate air or HEPA filters are used in operating theaters and laboratory laminar flow hoods to deliver sterile air, which is required in operation theaters. Excuse me, that is, the HEPA filters are used in operating theaters to deliver sterile air. We also have the use of radiation. There are two types of radiation: non-ionizing radiation and ionizing radiation. Non-ionizing radiation involves the use of ultraviolet light, which has low penetration or power. UV light is known to cause formation of thymine diamonds in nucleotides. It's also important to mention that UV light causes damage to the skin and eyes of humans. So it's important that UV lamps that are used in operation theaters, sterilization of work spaces, and tissue culture facilities should be switched on only after the operator has gone out. Ionizing radiation, such as gamma radiation, is used to preserve foods and also for sterilization of heat-sensitive medical materials such as gloves, dressings, syringes. It is known that irradiated foods have been in existence for a long time, but many people are a bit hesitant because of food safety and health. If I eat irradiated food, will it cause damage to my DNA? So it is required that irradiated foods must have a stamp to indicate that they have undergone radiation process. Thank you for listening.